Hello, everyone, and welcome to Advancing Neuroinflammation Research with iPSC Derived Microglia and Automated Assays for Drug Discovery. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Brainxell. To learn more about our sponsor, you can visit brainxell.com. We do encourage you to participate by submitting any questions you have during the presentation. To do so, you can simply type them into the Ask a Question box and click Submit. We'll also answer as many questions as we have time for at the end of the presentation, and you can also submit any technical issues there as well if you are having any trouble hearing or seeing the presentation. I'd now like to welcome our speakers, and for their complete biographies, you can click on the left side of your screen. Seth, you can go ahead and begin your presentation.
All right, thank you, Matt. Thanks for everyone for joining us today. I'm excited to get this kicked off and talk a little bit about our cells and how they're used in neuroinflammation research. So Brainxell was founded in 2015. We are a company that focuses on the development of iPSC-derived cells, specifically for the CNS. Everything that we do is in the CNS space, hence the name Brainxell. Our founder is Sushan Zheng, a professor from University of Madison, and he is now currently the director of the Center for Neurological Diseases at Sanford Burnham Prebys. And Sushan has really spent the last 30 years of his career focusing on the development of different cells of the central nervous system and finding ways to create these cells from stem cells. So if we want to take a look at neuro research, there's a lot of different in vitro models that can be used. For in vitro neuronal models, one of the biggest ones is really using rodent primary cells. So if any of you have ever worked in neuro and done in vitro work, you may have worked with mouse or rat primary cells. So you know the process of collecting and processing these cells and then coming up with a hopefully pure mixture of cells. One of the challenges here is obviously throughput. It can potentially take a lot of animals to generate very high number of cells, and you end up with often a heterogeneous mixture of different neurons. And then there's also a challenge here in terms of translatability. Obviously, if we are developing therapeutics for humans, using mouse and rodents isn't really a perfect model. But again, this is one of the most common approaches. Another option is human primary tissue. This is particularly challenging in neuro research, right? There is a tremendous limited availability of these cells. And really, the most human primary tissues are non-neuronal. You can get some neuro cells, maybe astrocytes or microglia, but for the most part, this is really a major lacking in the industry. And so finally, we have human iPSC-derived cells. So this is something that really has become more prevalent in recent years as the different cell types have become more available. This allows for an increased reproducibility, increased availability of the cells. The cost has decreased. And what this really allows us to do is focus on human targets and human pathways, right? So you can start from that from a fibroblast or a blood cell or even a urine epithelial cell and do reprogramming to turn that adult cell back into a pluripotent stem cell. So then that is an induced pluripotent stem cell. And then taking that stem cell and moving it forward and creating all sorts of different types of cells. So at Brainxell, we use a directed neuronal differentiation process. What this means is we are really trying to direct the cells from a stem cell state into a terminally differentiated cell type using very specific morphogens or small molecules, and using those at very specific time windows within the development, right? So you're starting from a stem cell, and for the beginning, you might be adding one small molecule and then another, and you're driving it down these different lineages, whether it is forebrain or midbrain or spinal column, and then further differentiating into those specific subtypes. At Brainxell, we have many different types of cells, from cortical glutamatergic to cortical GABAergic to striatal medium spiny, spinal motor neurons, and also midbrain dopaminergic neurons. So these are very pure, very subtype specific neurons that are generated, again, from stem cells. And we can do the same to make the glial cells. So things like astrocytes, we have both spinal and cortical astrocytes, and then also microglia. And then a new cell type that we're coming out with is oligodendrocytes. So these are different from neurons, and they are often used as, these are support cells or the immune cells of the nervous system. And the goal here is to really produce cells that are relevant for specific diseases. Different neurological disorders such as ALS or Alzheimer's, multiple sclerosis, these all have different cell types that are affected. So in the case of ALS, these are motor neurons, and then spinal astrocytes and microglia are really relevant for that disorder. Something like Huntington's disease has a very specific cell type, the striatal medium spiny GABAergic neuron. And one of the cells that really kind of crosses all of these can be microglia, because these are a neuroimmune cell. And so the disorder that somebody is working on can really dictate which cell type they want to be using, whether it's motor neuron or glutamatergic or dopaminergic. And then beyond that, there is the ability to also create disease models. So disease models can be either generated directly from a patient line. So a patient who has a disorder with a known mutation may donate their fibroblasts or their blood cells that can then get turned into a stem cell, and then that stem cell can get turned into the specific disease type or the neuron of interest. But that's not always the case. You can't always get access to patient samples, and sometimes the best course is to start with a control line. So something like a healthy, non-disease control line that has a normal genotype. You can take that iPSC and have it edited to create a very specific mutation. So in the case of Alzheimer's disease, you may want to generate an ApoE4E4 mutation, or a TREM2 R47 SNP, or an APP or presenilin or MAPT mutation. And all of those can then be taken and converted into the different specific cells that are going to be very relevant for that disease. And so that's where we kind of start here is in the case of this seminar, we're starting with microglia and taking an iPSC line and having it edited. And this is a perfect segue for me to hand over to the next speaker, Travis from Editco, who does all of the editing.
Great. Thanks, Seth, for the introduction into Brainxell and all the capabilities that they're able to do. I promise not to belabor this part of the talk that much longer, as I know everyone is probably here to see the exciting data that the Arctoris team is going to present. but I would be remiss if I just didn't take a little bit of time to talk about Editco and who we are quickly before we dive into that. So, with that said, who is Editco? If you're new to the genome engineering space, or if you're not new, we might be a new name for you. However, Editco in its current form has actually been around since about 2018, but it was under the umbrella of Syntheco. In early 2024, the engineered cells and multi-guide reagents business actually spun out of Syntheco to form this new entity, which is called Editco, which is who we are now. Since our inception in March of last year, we've actually been able to expand our capabilities beyond editing just memorialized iPSC cells, or iPS cells, but into the primary cell editing space with our T cell, and primary fibroblast editing portfolios. And we're looking forward to some many new and exciting product launches that will come later on this year that I'm sure that you guys will hear about and we'll talk about and shout about from the rooftops. But with that, as you can see, we actually have quite a bit of a legacy and experience in the genome engineering space, as seen by some of the numbers on the screen. We've been responsible for well over 500,000 genome edits when you take into account both our services and the reagents that we've been able to sell within the market within our portfolio. Additionally, we've been cited in, I think at this point, well over 700 publications, especially when you take into account the products and services that we brought over from Syntheco whenever we spun out. And from a cell line engineering perspective, we've been able to fine-tune our automated platform to have an average edit efficiency for our pools of well over 80%, which has allowed us to put in guarantees for our XTEL cells, which was recently launched earlier this year, to have that 80% editing guarantee in our pools. And as you can see by the number on the screen there, we've worked with well over and approaching probably closer to 700 different cell lines at this point. Additionally, we've worked with and collaborated with many of the top researchers in the world, as seen by some of the logos on the screen there, such as St. Jude's, UCSF, and Citro, Brainxell, and MD Anderson, and some of these other companies that are on there. So what do we offer? So what does our product offering look like? As you can see, we offer a full range of CRISPR products and services, ranging from our gene knockout kits for those customers who would like to perform their genome editing in their own hands, to a full suite of edited cells in both memorialized iPSC and both pools and clone formats, as well as knockout pools and primary cells such as T cells and fibroblasts. So at the core of Editco, what we're really focused on is democratizing CRISPR, right? Editco's mission is to make CRISPR technology accessible to everyone, from these large research institutions to some of these small biotechs and even the academic individual researchers in the world. We aim to do this by offering an affordable, sort of easy to use CRISPR kits and cell line engineering services that are as well as our comprehensive sort of technical support and customer support teams. We're really aimed at removing the barriers for those that are traditionally limited to the use of this powerful technology, such as CRISPR. For engineered cell services, we achieve this through our CRISPR platform. This platform is a groundbreaking CRISPR editing platform that aims to deliver the new gold standard in cell-based models. So researchers truly do leave no variant, gene, or mutation unstudied ever again. Yeah, what we have on the screen there is a bit of a peek behind the curtain of our highly automated factory, which is based out in Redwood City, California. This is what I like to call a bit of our cell nursery. The images on the right there is actually where all the outgrowth and expansion of our cells take place in a highly automated fashion. Following single cell printing of clonal isolation, the cells are actually moved around in a module in a large plate format, and images are actually taken every 2 to 3 days. We screen an average of 400 clones per project, and as you can see on the images, the amount of data and images add up quite quickly. And so we have a huge shout out to our data team and our software team for putting this automation in place and designing these intricate systems so that we can monitor projects from front to back and make sure that we have high quality cells. Again, not to belabor this too much longer, and I won't dig into too many of the details, but just focusing on iPS cells, as this is what a lot of this talk is about. I just wanted to provide a quick overview of what that process looks like from our standpoint, from our platform standpoint. From beginning to end, we have a comprehensive team that monitors and takes care of all of these projects, from the helping design with the editing strategy to the comprehensive QC that we're able to provide on the back end of a lot of these projects. And just to dig in a little bit deeper on our iPSC offering, as this is what we're talking about, with some of these projects that Arctoris is going to do, we do offer a comprehensive suite of knockouts, single nucleotide variants, as well as tag inserts in our iPSC cells. And one of the things that we know is important to anybody who's working with iPSCs is maintaining a stable carrier having pluripotency. So we do offer additional services to researchers, such as pluripotency assessments and genomic stability assessments that are add-on services that we can provide, so that we can provide that extra confidence that the cells that you're getting are fully high-quality cells with the edits that you need to perform your research. And last but not least, before I hand the talk over to Chris from the Arctoris team, I just wanted to state, as Seth said earlier, we can work with patient-derived cells and do point mutations or mutations back to the wild type, but we do have a full suite of control cells on our platform that we've already optimized and work with quite frequently and have optimized protocols for. And of those, at the bottom there, you can see that we have partnered with Brainxell to create working banks or starting banks of both their BX30 and BX32 cell lines to initiate projects immediately. But with that, as I said, I don't want to go on too much longer. I want to be able to pass this off to the Arctoris team to provide some of the great data that they have with some of these cells.
Thanks very much, Joris. Perfect. So, yes, forced me just to introduce Arctoris as the final part of this this collaboration before I hand over to Nero, really, who is the person that's going to be taking you through all the exciting data that we've generated. So Arctoris is a tech-enabled CRO, and what that means is we bring automation to the laboratory. What you can see on screen here is Ulysses, which is our modular cell biology platform, looking fabulous in night mode, I have to say. The platform here allows us to really tailor our approach to specific client questions. We can take everything from basic cell culture, feeding, seeding of cells, right the way through complex assay design, and into multiplex readouts such as flow cytometry, plate-based readers, and high content imaging. Now, we are based in Oxford in the UK. We're almost 10 years old as a company, and we've got expertise across a wide range of areas such as biochemistry, cell biology, biophysics, protein and structural biology. But everything that we do really is focused on data quality, reliability, robustness. We believe that is the most important thing during the drug discovery process, to make sure that you've got confidence in the data you're generating. And why is that so important? Well, there is currently a reproducibility crisis in science. There's some pretty depressing statistics out there, such as the fact that half of scientists report that they are unable to reproduce their own work. And this rises further to more than 70% of researchers failing to reproduce work that has been published in the literature. And these statistics then end up with things like an attrition rate in neuroscience drug discovery that's estimated to be around 90%. Now, this means that 90% of drugs that make it through to phase 1 clinical trials fail to become approved drugs at the end. So how do we go about improving this? Well, at Arctoris, we believe that automation is a key way of improving these kind of statistics. We believe that manual methodology cannot cope with the demands of modern drug discovery. We think that higher quality data is required to train AI and machine learning models. And as research costs increase, this higher data quality allows you to make earlier decisions with more confidence in your projects. The benefit of the automation means that our assays perform the same today as they will next month, as they will next year. It doesn't matter if it's 8 AM or 8 PM, or if the operators had 1 copy or 5 copies in a day, our assays will perform the same. And that's more than I can probably say for some of our scientists. At Arctoris, then, we combine scientific excellence with high-end automation. It allows us to produce superior data quality, increase robustness and reliability, and we have consistent performance at scale over time. but there's no point in generating fantastic data using the wrong model. And this is therefore where this collaboration with Brainxell and Editco really came from, is that we think the correct model is a key consideration in drug discovery. As Seth mentioned earlier, primary human brain tissue is difficult to access, so a lot of drug discovery in neuroscience has been done on cell lines and primary rodent cells. We know that these have translational challenges. We know that they are potentially not physiologically relevant to the disease population that you're trying to treat at the end of the day. So as our push for higher data quality, we believe that the right model should be a key consideration. And the iPSC space has really exploded in recent years, meaning that now these are a genuine alternative to these traditional models. So Brainxell have shown that you can generate human and patient-derived samples and iPSCs at scale and generate reproducible cells that we can use on our platform in drug discovery. And that's what the talk is going to be about today. So with that, I'll hand over to Naroa. Really, what we have today is two stories. One is around the onboarding of an inflammasome assay, which shows kind of how these cells and our automation together can start to generate assays that are unachievable by manual methods. And then the second story will be around an edited line from Editco, some characterization to show how we can incorporate them into the platform and drug discovery as well. So with that, I'll hand over to Naroa, fantastic neuroscientist, fantastic senior scientist in our team, with a lot of experience in iPSC biology and neurodegeneration, and really the driving force behind all this data that you'll see today. So with that, over to you.
Thank you, Chris, and thank you everyone for the wonderful introductions. And as Chris has said, now I'm gonna talk you through a project we've done to kind of bring all three companies together and to develop an assay in-house to study inflammation in microglia. So I'm gonna start with a brief introduction to why are we interested in studying neuroinflammation, how is it related to disease. So first of all, in the last few years, it's been established that neuroinflammation is a hallmark of neurodegenerative diseases such as Alzheimer's and Parkinson's. And there's many different ways how this inflammation affects the health of the brain. One way is that excessive glia activation, microglia, astrocyte causes damages to the neuron, and also these activated glia cells, they lose their homeostatic properties towards these neurons. That's why in the last few years, a lot of researchers have pushed their focus towards these glial cells as a therapeutic target to try to dampen neuroinflammation and that way try to treat the diseases. For the purposes of this presentation, we are going to focus on microglia, iPSC Derived Microglia, which were provided by Brainxell, and in the field of Alzheimer's disease, because there is a lot of evidence that has shown how microglia and altered activation in microglia are involved in Alzheimer's disease. Because, for example, these microglia are responsible for taking out and degrading all these amyloid plaques and toxic protein aggregates that are found in the brain. But when they become activated, this degrading property of the microglia is impaired and it causes damages to the neurons. So inflammation can happen in many ways in the brain, and one such very important pathway is called the NLRP3 inflammasome. Inflammasome are very big, you can see them here in the right, big protein complexes that are found in the cytoplasm, and they mediate very robust inflammatory responses to a range of different cellular stresses and infection as well as disease. So this, as you can see in the image on the bottom, there is a range of different inflammasomes, but the most characterized one and the most important one that we think is the NLRP3 inflammasome. This protein is widely expressed in the central nervous system, but its stronger expression is in microglia. And this protein, or this complex, and its signaling pathway has been involved with many different diseases, such as Alzheimer's, which we'll discuss now. There you go. Indeed, this NLRP3 inflammasome has been linked to Alzheimer's disease, and there is a lot of evidence to back up this claim. One of them is the products of the signaling pathway, interleukin-1 beta and interleukin-18 have been found to be elevated in Alzheimer's disease patients. Then amyloid beta itself, the toxic protein involved in Alzheimer's, can activate this signaling pathway. Then the interleukins and amyloid beta can act in a feed-forward loop. And very importantly, it's been found that knocking out this NLRP3 protein can improve the clearance of amyloid beta in animal models. That's why this NLRP3 inflammasome has been determined as a target, as a therapeutic target to treat Alzheimer's disease. So how does this NLRP3 inflammasome work? What's the signaling pathway? How do we activate it? So two signals are needed to activate the NLRP3 inflammasome. First of all, we need a priming signal, which occurs by activation of the TLR receptors or in cytokine receptors. Then the NF-kappa B signaling pathway activates, and this leads to increased expression of the NLRP3 protein in the interleukin-1 beta and interleukin-18 precursors. And then we need a second signal, which is called the activation, which it can happen through activation of a range of different receptors with different molecules. And this leads to formation of the inflammasome complex in the cytoplasm, activation of caspase-1, and this caspase-1 protein then cleaves the interleukin-1 beta, interleukin-18 precursors, and also gasdermin-D, which then forms a pore in the cell membrane that causes the release of the cytokines. So an issue with this sort of double activation, double step activation is how do we do with this in vitro? Because indeed, as you can imagine, both signals need to happen pretty close in time. So if we want to do this in a lab, we need to very precise timing of the treatments. Also, if we want to include compounds in between both, that becomes quite challenging to do manually. And also, if we have a cell model that is not reproducible, that has a lot of variability from well to well, from plate to plate, from experiment to experiment, this assay is very, very hard to reproduce. And in the literature, in the past, most people that have studied NLRP3 inflammasome have focused on interleukin-1 beta secretion. However, this cytokine can also be produced through different signaling pathways, so it can lead to false positive because it's not only the NLRP3 inflammasome that is going to lead to an increase in this cytokine. So what we've done in Arctoris with this assay, we've done using automation. It has allowed us to do these treatments in a very precise way in a quite large scale, and also using the the microglia, the iPSC Derived Microglia provided by Brainxell, we can do this in a very robust cell model that is reproducible across plate and across experiments, and also it reduces the timing of the experiments a lot because from the time of thawing to data collection is just 1 week or less. And at the same time, using automation, we've managed to multiplex 2 different assay readouts because on top of the interleukin-1 beta secretion, we can also measure caspase-1 activity in the cells. And this will confirm that the results we are seeing, such as activation of the NLRP3 inflammasome, it will validate them in 2 different readouts and reduce the number of false positives we may have. And it also a platform that will enable us to study compounds that will inhibit the inflammasome. And down here, you can see the example diagram of the approach we use for the treatment. So now I'm going to guide you just briefly, so onboarding with it to develop this assay, to optimize it so that then we could increase the scale to bring it to high throughput so that it will be ready for compound screening using automation. So the first thing we did is what do we use to activate the inflammasome? So we said, okay, for the priming LPS, and then for the activation, either ATP or nigericin, we tested different concentrations. You can see here in the top right an example matrix assay. We tested different concentrations of the different compounds. We tested different incubation times for each of the steps. We also compare ATP or nigericin to see which one will be better in the activation step. And as you can see in the bottom right graph, at least in our hands, it appears that activation with LPS and nigericin, it gives a bigger activation of the inflammasome than LPS and ATP. This means that our assay window will be higher with nigericin, and thus when seeing if any compounds reduce this activity, it will be better to have this higher assay window. We also tested an inhibitor, which I'll talk more through in the next slide. Because in order to have a positive control, if we are looking for compounds that inhibit the inflammasome, we need to have a control that we know will inhibit the inflammasome. And what such compounds is MCC950, which is a very specific inhibitor of the NLRP3 inflammasome. And we tested a range of different concentrations, and we found the most optimal one that we use as a control for our subsequent experiments. And also in this assay, it enabled us to see that DMSO in the concentrations used was not interfering with this signaling pathway and with the NLRP3 inflammasome. And lastly, as an onboarding, once we had our treatments, concentrations, timings, all set up, we said, like, okay, how can we use our automation platform? How can we use this assay in that format? Is it reproducible? Is it robust enough? So we did a first run, similar to what we will then do for our screen, but just using our high and low controls. That is stimulated cells with LPS and iGericin, just with DMSO, or also with the inclusion of MCC950. And on the right-hand side, you can see some of the different liquid handlers we've used for this automation. And as you can see in both of the graphs, this assay, the controls work really nicely. We have consistent responses across different wells, and the controls are doing the way, behaving the way they should. So next we did, once this assay is set up, let's do a screen of compounds. So what we did just to test that this assay is good for compound screening, which can then be used in the future for different projects with a large scale of compounds. But we started just with a relative, the mid-scale one with 246 compounds that was distributed across 3 plates, which were running parallel, which enabled us to see how reproducible results were from plate to plate. Each compound was running technical triplicate to see if there was any variation across the same plate. And we also use a high sample number of high and low controls. And as I mentioned at the beginning, we did a multiplex readout of these same samples with caspase-1 activity and also with interleukin-1 beta using an ELISA, just to make sure that those results were seen, or like positive or negative hits were actually true and not false or true or false positive. So we ran the screen, and before we went on to look at the compounds, we focused on the controls to see whether the assay had performed in an effective way, whether it was robust enough, whether there was any variability between the plates. And as you can see here in the top graph, all 3 controls are called all 3 plates behave very similarly, which was great. And also the interassay coefficient of variance percentage was only 12.6, which is quite good. Then the signal to background ratio also was good, giving us a good assay window. And lastly, the coefficient of variance is of the treated cells is relatively good too. So we decided, okay, the assay seems to have performed well. There is not a lot of variability across the plate, across a single plate, or across the different plates. So then we went on to the data analysis. This is the most interesting part. So in this first graph, there is a lot of dots, I know, but the data that is shown here is the caspase-1 activity relative to the unstimulated cells. And each dot is the average of the technical triplicates of each compound. And here, the blue line one is just unstimulated cells. Then our yellow line is those cells that have our control MCC950, and the pink line is LPS, cells with LPS and iGericin, our kind of high control. And things you can see is, first of all, our compound library has a number of compounds that inhibit this inflammasome activity because their dots are below the pink line, which is great. I should mention that this compound had, this library had some of compounds that we knew were going to inhibit the inflammasome, and they did behave that way, which also gives more credibility to our results. And lastly, there were also a number of compounds that were increasing the inflammasome activity. And this finding is very interesting in itself, and it's also important because it indicates that our treatment is not overloading the microglia, meaning that we are not working at the very higher end or highest end of the activity of the inflammasome in this microglia, because its activity can even be higher increased. So the microglia are not in an extremely stressed state in our activated state. So using this in samples, we did our next assay, which was the interleukin-1 beta ELISAM. And the graph is the same, it's just showing, in this case, the interleukin-1 beta levels as a percentage to the control. And once again, we have a lot of compounds that seem to lowering these levels of interleukin-1 beta respective to the high control, whereas there is a few compounds that also are increasing this release. So something we did next, the most is compare both results for all the compounds, which is the graph here. The x-axis shows the caspase-1 activity, and the interleukin-1 beta, and the y-axis shows the interleukin-1 beta release. And as you can see, overall, there is a very nice correlation. And so, for example, all these compounds in the bottom left quadrant, most of them will be inhibitors of the inflammasome, whereas those in the top right quadrant will be activators of the inflammasome. And those in between can be either false positive or maybe another different pathway has been activated. But yeah, overall, we are very happy with this, how this screen went. And it will be very interesting to then follow up with some of the leads. Yeah, so conclusions. First of all, the microglia we've used by the iPSC Derived Microglia used by Brainxell are very robust. They are giving us a lot of reproducible results across different plates, across different experiments. They are very good and a human model to be used in vitro and to study this inflammasome pathway. Then combining these cells with our automation platform in Arctoris, we've managed to develop a screening automated assay platform to screen compounds in an assay that has multiple addition steps in a very short period of time. And it enables us to get a lot of data in just a short amount of time. And again, it enables us to multiplex 2 different readouts, which validates the different hits we may find. And as next step, what we want to do now with this assay, with these cells, is first of all, increase the complexity of our model, because we know in the brain, we don't have microglia in themselves, we don't have neurons by themselves. So we will be very good to combine this to develop this assay into a co-culture system, to a more complex neural system in vitro. And the next step, all the experiments I've shown so far were performed in 3A4 well plates. So our goal now will be to lower the scale and increasing the throughput by doing this assay in 1536 well plates. This will decrease the time needed to screen compounds and increase the data we are using. And we've done some preliminary assays by growing the microglia in these 1536 well plates, and we've gotten very promising results so far. So we'll be excited to see whether we can do this in the future and do these very high throughput screens. And now we are going to discuss how not only using wild-type microglia, but we can also do assays in microglia that carry our mutations of interest, mutations related to a specific disease. In this case, the train2 R47H model, and this brings all 3 companies together as Editco edits the iPSCs. Brainxell differentiates these genetically modified iPSCs into our cell type of interest, and then we've done assays with them. And Chris is going to give us a brief introduction into iPSC editing.
Yeah, thanks, Nora. Really good talk so far. Give you a chance to get your breath back in a second. So basically what we're looking at here now is trying to take the value of iPSCs that one step further. So using iPSCs provides us with this human model. But the real value here is that we can start editing these. We can start bringing in a specific target of interest mutation in here. We can start bringing in a specific disease relevant mutation, such as the TREM2 R47H mutation that we're using here, and really start to look at doing your drug discovery program from day one. So your initial screening through your iterative design, all your chemistry is then done on a disease relevant line, so that when you get to clinic, there's fewer surprises for you, because you've essentially kept your target population in mind throughout your drug discovery process. So that's going to be the second part of this talk. It really is just showing that assay onboarding that we've just done with this assay isn't a separate story here. we can start incorporating these iPSC edited cells into our workflows and really increasing the power of your drug discovery campaigns from day one. So hopefully a nice little break for you then, Nero. Get your breath back and yeah, dive now straight into some discussions around this this edit that we've done with Editco. Back to you.
Thank you, Chris. Yeah, so this part of the presentation, as I said, we are focusing on microglia that carry a TREM2 specific mutation. And Seth said during his introduction how this TREM2 gene is related to Alzheimer's disease. But what is TREM2? What does it do in the cells? So TREM2 encodes a receptor in the microglia, and the signaling pathway that is activated when this receptor binds to its ligand, it's involved in many of the processes of microglia, such as phagocytosis, proliferation, survival, and it's been shown that TREM2 has a protective effect in inflammation, as it suppresses the pro-inflammatory state of the microglia. But what happens in Alzheimer's disease? So it's been shown that mutations in the TREM2 gene are a risk factor for Alzheimer's disease. And an interesting fact is that amyloid beta, this toxic protein involved in Alzheimer's disease, binds to the microglial TREM2. It's also been shown how deficiencies in TREM2 result in higher accumulation of this toxic protein, higher phosphorylation of these toxic proteins. And that's why a lot of people are using this TREM2 as a target to develop therapies, and some are actually entering clinical trials. And lastly, the mutation we are going to be focusing on is the R47H mutation, and this mutation reduces the binding capacity of the TREM2 receptor to some of its ligands. So this nicely puts together all our different companies, because Editco, as we mentioned, they were the ones that have generated the iPSC lines that carry this TREM2 R47H specific mutation. Then Brainxell has differentiated these iPSCs into microglia, and the microglia will still carry the mutation because the genome is the same. And they've done some characterization in the cells, and they've shown how there is these TREM2 microglia have some differences with the wild type line, whereas other things are still the same. For example, these TREM2 microglia have lower adherence, they are smaller inside, and less ramified morphology. But for example, when looking at the expression of different genes, they don't have any differences in IBA1 expression, but for example, yes, in TMEN119. So it's really interesting that there are some different genotypic differences, but not others. So one step further, here in Arctoris, we also did some characterization of the microglia, of the TREM2 compared to the wild-type microglia. And we saw here, top on bottom left, in a cell viability assay, we didn't really see any differences between the genotype. However, in a phagocytosis assay, this TREM2 microglia seem to have a reduction in the phagocytosis of amyloid beta, but not in the S aureus particle. And also, there doesn't appear to be any difference in the phagocytosis when stimulated with inflammatory cytokines. However, a difference we are starting to see is in the response to the inflammatory stimuli of these TREM2 microglia in different pathways. For example, we've also done this caspase-1 activity assay in both wild type and TREM2 microglia, and the TREM2 appear to be more sensitive, meaning that they have a higher response, a higher caspase-1 increase in caspase-1 activity when we stimulate them. And on the right hand, you can see different graphs that are cytokine release in response to either LPS of amyloid beta. And once again, in the pink line, you can see how the TREM2 microglia, the cytokine release is higher than in the wild-type microglia, which is really interesting. And we will do more assays in the future to explore this further. And it will be also very interesting to do these compound screens, both in wild-type microglia and in TREM2 microglia, to see how this mutation can also affect how these compounds interact and affect the TREM2 inflammasome. And I've been talking enough, so I'm just going to pass it over again to Chris so that he will give us just the last conclusions.
Perfect. Thank you very much, Nora. Yeah, really whistle-stop tour of the kind of characterization that we'd be doing on these cells. But what's really interesting is the ability now to be able to incorporate this kind of thing early into a drug discovery campaign. So in conclusion, what have we spoken about today? Well, we've shown that Arctoris' automation platform can improve data quality. It allows us to scale methodology to levels unachievable by manual methodology, and generate data on assays that traditionally would be very hard to scale at this kind of level. We've shown that Brainxell iPSC derived microglia are suitable for scaling. They can be used in this screening kind of work and have really good reproducibility across different experiments, different plates. And finally, the Editco's editing platform allows iPSCs really to be edited for inclusion in drug discovery at a very early stage, whilst also providing insight into the different roles of specific targets. And really what we're trying to achieve here with this collaboration is showing that each of the three companies that are experts in their own respective fields coming together can actually offer something unique in this space. And just really to leave with a final kind of thought, as the FDA announces this week that they are starting to phase out animal testing requirements, there's probably no better time to start thinking about including iPSCs into your drug discovery pipelines. So with that, final acknowledgements. These are the brains behind everything that you've seen today. And then a special shout out really to Ulysses at the bottom there for doing all the work at the weekends and evenings so that our scientists don't need to. And with that, thank you very much. I think Matt.
Yes, thank you very much, everyone, for those wonderful presentations. What we'll do now is we'll go into the Q&A portion. So let's start with, and then you all can come back on camera and unmute as you answer there. Can you do this same assay with neurons as a co-culture?
Yeah, I can take that. It's certainly something that we could do. It's something that we'd like to look into. As kind of Nero mentioned in the talk, these things don't exist in isolation in the brain, so it would be really useful to have additional cell types in here. We could take that further and start looking into potentially even tricultures and quadcultures as well. What would be really cool about that is you could start incorporating different readouts in there as well. So we could look at NLRP3 inflammasome, we could look at inflammation, for example. We could also then start looking at neuronal readouts. So we start treating microglia and read out neural outgrowth reading from neurons, for example. So you can start treating one cell and reading out from a totally different cell, which is a really powerful tool to be able to use.
All right, thank you. The next one we have here is, how long does it take to make the microglia?
Yeah, so obviously I can take that one. So there's kind of different ways to think about the making of the cells. You know, if somebody was to send us an iPSC line, our general statement is it takes us about 4 to 6 months to receive cells to the time we can deliver them. However, that also kind of depends on the quality of the iPSCs, you know, if they have to be cleaned up. If you're starting from fibroblasts, that's going to make it longer because you have to reprogram. But then also, you know, if, for instance, if we're starting with our line, we can make the microglia in about a month's time, and then there's just additional QC time. So the whole span of it can kind of depend really on the quality of the cells coming in, and then also, you know, any unique situation within the cells.
All right, thank you. The next one we've got here is, are you using an inducible system so that the GOI could be induced or KD after the differentiation?
Yeah, so I can take this one as well. We do a fair amount of inserting into like an AAVS1 site, and that can certainly be an inducible system. We use a TET2 system. to turn on genes of interest. You can also, overexpress. You can basically insert so for instance, we have a GFP line, we have GCAMP lines, RCAMP. So these are all endogenously expressed, but then you can use a inducible promoter and turn something on whenever you want. You can also utilize, for instance, if you want to do a knockdown, lentivirus works really well in our mature neurons, and we've actually got protocols for that type of stuff. But everything is kind of on the table. Usually, if you're doing like a gene insertion, we're going to do that at the iPSC stage, and then do the differentiation into the microglia or the neurons or whatever cell type.
All right. Let's see, the next one we've got here is, can you build a complex model, say, of Parkinson's disease using automation to test drugs against?
I'd say that yes, basically. So the technical aspect there, if you were looking at Parkinson's, say you'd want maybe a triculture system, something like that. The technical aspect here is creating consistent media across the various different cell lines. Brainxell have models like that. It's something we are actively looking at bringing in into Arctoris. They've got a triculture of neurons, astrocytes, and microglia, and I now believe they're also working on the quadculture system, which we'd like to get our hands on as well, including oligodendrocytes. From our perspective, it's essentially an assay transfer piece where we have a lot of experience in bringing complex models onto the automation. So it would basically end up being a case of just optimizing how we do the seeding with the automation, how we then continue the feeding to make sure that the cells are healthy throughout. But it's eminently feasible and something that we are actively exploring. What's really exciting then, I guess, is that you could take that triculture system, quadculture system that you've got automated, and just start substituting cell lines in there for specific edited lines. So you could take a specific mutation of interest, you could take a specific disease relevant line, and see the impact of that cell on the quadculture or triculture system. So that's something that could be really powerful. The benefit of the automation in the iPSCs is actually you could start using these models probably earlier in drug discovery than traditional. These don't have to be a very late stage thing anymore. You could start doing that from a very early standpoint.
All right, excellent. The next one we've got here is, what is the upper limit of number of compounds you can test in a single assay?
Yeah, I can't give you an exact number. It would partly depend on the complexity of the assay, the timeline of the assay. So it's then basically a logistics question of how the automation could fit things together. We have projects running at the moment that screen 5,000-10,000 compound libraries in here. It's not too challenging to scale that. With the automation, this can just run 24 hours a day for however long it needs to. So yeah, I think at the moment we have, on a different model, a 1536-well assay screening 5,000 compounds in the space of 3 or 4 days, I think. So that kind of level. We could go higher. We could run 10, 20,000, depending on the specific assay of interest.
All right, we've got time for a few more here. The next one is, do you have DMSO or other solvent tolerance data?
For this assay, we do, as part of our standard onboarding, we'd always look at DMSO tolerance for when we start to add compounds into the screens at the end. I don't know what the data is exactly off my hand here, but essentially we'd be able to use DMSO concentrations up to a sensible volume to be able to do the actual screen. So I think it's something like less than 0.5% we're using here, but these cells can probably tolerate higher. It's something we do as standard, and yes, each assay would then have a maximum DMSO tolerance that it could cope with.
All right, looks like we've got time for one last one here. I apologize for some of the pronunciation here. Do you have an explanation for the difference between the S. aureus and the B. amyloidoid? You know, I'm going to let you, if you want to look at that question there. Yeah, if you want to read that one out there, I'm just going to mess that up because there's a couple ones I didn't have a chance to check before the thing today.
No worries. So the question is, do you have an explanation for the differences between the Staph aureus and beta amyloid phagocytosis in the microglia, wild type versus TREM2? Is there a specific mechanism of action? It's a very interesting question. There is a thought at our side that because TREM2 is known to be a recognition receptor for amyloid beta, that potentially the microglia just aren't able to recognize it quite as well, and therefore we see that reduction in their phagocytosis of amyloid beta. And there's likely different pathways involved with Staph aureus phagocytosis. It's not something I can definitively say, and there could be other factors at play there, but that is our working hypothesis at the moment, that it's basically different pathways involved in the phagocytosis. It's something that will be interesting to pursue further, but it builds into this whole narrative of using the right model and the right approach. If you are wanting to improve the phagocytosis of amyloid beta for Alzheimer's disease, for example, if you're using Staph aureus as your bait, are you actually assessing the performance of your compound on the right model? So it all builds into this whole idea of you can have as brilliant a data as you like. If you're using the wrong model, can you actually rely on what you're generating? So yeah, really interesting question.
All right, excellent. I want to again thank our presenters for their time and important research. I want to thank LabRoots and our sponsor Brainxell for underwriting today's educational webcast. Before I go, I want to thank the audience for joining us and all the questions. Questions we didn't have time to get to today, and anything that is submitted during the on-demand period will be submitted to the speakers and followed up via the registration information you provided. This will be available on demand. LabRoots will alert you via email when it's available for replay. And we do encourage you to share that with any colleagues who might have missed today's live event. So until next time, thank you very much and goodbye.